I'm George Stamas, and on behalf of my fellow directors and executives at the Hellenic Initiative, we want to welcome the many hundreds of you from Zoom and Facebook who have joined us today for this extraordinary inaugural THI Digital Speakers Series. And there cannot be any more timely speaker event than to welcome Doctors George Yancopoulos and Christos Kyratsous of Regeneron. I think, as all of you listening know, these two gentlemen and their company, Regeneron, are at the epicenter of the fight against COVID-19, racing and leading, really, the pharma industry in the research for an effective vaccine. And the world eagerly awaits daily announcements from Regeneron on their progress. So what does all this have to do with THI? Actually, everything. We at THI celebrate the achievements of people like Dr. Yancopoulos and Kyratsous, and they inspire us to do our part to improve the lives of the Greek people with our many programs and our recently launched social media campaign, which is raising funds at the grassroots to address the health and welfare issues brought on by the devastation of COVID-19 that we expect will have such a devastating impact on Greece's tourism season this summer. Tourism in Greece accounts for 35% of Greece's employment and its GDP. And even at the worst depths of the recession 6-7 years ago, which launched THI, there was still a tourism season. And we worry very much about this year. Dr. Kyratsous is Regeneration's head of infectious disease research, leading the development of antibodies against COVID-19. Christos is a Greek citizen with advanced degrees too numerous to mention from Aristotle University in Columbia. And he's celebrated worldwide for developing a MERS treatment in 6 months, and then the best Ebola drug that exists. And now he has taken on what might very well be the biggest challenge of his career, finding a cure for COVID-19. Dr. Yancopoulos is co-founder, president, and chief science officer of Regeneron. He grew up in Woodside, New York. He was valedictorian at Bronx High School and Columbia. And he holds 100 patents himself. He's the principal inventor of Regen's 8 FDA-approved drugs and its other foundational technologies. Under George's leadership, Regeneron has grown to a current market valuation of $63 billion. And more importantly, he would say, I'm quite sure, what he has done is improved the health and welfare of countless people, for which we are so grateful. The world owes Regeneron and these gentlemen a huge debt of gratitude for their service to mankind and their dedication to making the world a better place. And for us to have 1 hour of their time today, in light of everything they're in the middle of, is truly remarkable. We're humbled and honored. Welcome, gentlemen. I'd like to start, if I can, by introducing Chris Goulakos, who is one of the leaders of our new leaders program at THI, and Peter Poulos, our executive director. And maybe I can lead off, George and Christos, by asking you a little bit about your family backgrounds, your connections to your Greek heritage. I think everyone would find that very interesting, especially so many of us are from the Peloponnese, which you're not. So please, George.
Well, first of all, thank you so much for having us. And I think you'll hear from both of us that in many ways we owe everything that we are and everything that we're doing to our Greek heritage. So it's an honor and a privilege to be able to share that background and that experience with you guys.
Thank you.
Both my parents come from what was then a small town in northern Greece called Kastoria. My father served in both with the British forces in World War II, and then afterwards in the Civil War in Greece, in northern Greece. His education was interrupted. He never finished school, never finished high school. And he managed to bring his whole family over in the late 50s to Queens, where he managed to reconnect all these people who had been dispersed and separated from the war. And from the day I was born, he would emphasize to me that though he was struggling, lack of education, coming from war, having to take care of countless people that he was trying to take care of, he emphasized to me that in our history as Greeks, we have greatness. We have so many examples of heroes who have done so much, from the ancient times to the modern times. And he told me, he also gave an example of my grandfather. My grandfather had been educated, and that's a whole long story that I don't think I have time to get into. And he said, you know, you can be like your grandfather, you can be like these other Greeks, and you have that heritage to live up to. And all you have to do is work hard and study hard, and you can achieve something. And he raised me on those beliefs and on how important my Greek heritage was to me, and how, you know, that he believed that Greeks had something special in us that we could really achieve and contribute to the world. And then in the mid-1970s, when I was getting more serious into my studies, he started realizing that I wanted to become a scientist. And believe it or not, he was disappointed in that, because we were not well off, and his dream was education was going to lead me to a productive and successful career where I could make money. And he would have been very happy if I became a real doctor, as he put it. But becoming a scientist to him was going to leave me working endless nights in the laboratory and making no money. So he, like many Greeks of that generation, he never fully assimilated, and he was still reading the Ethnikos Kyrikas every day. And I'll never forget, and I still have an article from the Greek newspaper that he cut out for me when I was telling him that I now want to become a scientist. And it was an article about Roy Vagelos, and the year was 1975. And the article was about Roy Vagelos being a famous academic scientist, which my father didn't have that much regard for, and leaving academia to go become head of R&D at Merck. And my father says to me, you know, if you want to be a scientist, don't be like any scientist. See this Greek scientist over here, Roy Vagelos, be like Roy. And over the next 10 to 20 years, many of you know, Roy really changed the course of biopharma research. He revolutionized it, and now he is recognized as the father of the modern biopharma industry. And along the way, for those 20 years, my father would cut out every article in the Ethnikos Kyrikas about Roy, and there were lots of them. And he would say, this is the guy, be like this guy, be like this guy. And so in addition to giving me, you know, the ancient heroes from Alexander and Aristotle and so forth, he gave me a modern Greek hero. And it turns out that the great Roy Vagelos, like I said, he is, for all of us who work in the biotech space, who are trying to come up with new medicines to treat difficult diseases, the great Roy Vagelos, another first-generation Greek immigrant, has set the standard and was the role model. And it is no coincidence that so many Greeks are now leaders in our industry of my generation. So you can look at the head of Pfizer now, you can look at the head of Biogen, of so many other companies, so many Greeks are so involved at the highest levels of so many of these companies. And it's because so many of us saw Roy Vagelos as a modern day Greek role model and Greek hero. And I'm not going to give all the details of it, but my whole life, my dad was saying, you know, why don't you just call up Roy? You know, you're trying to do this, you're trying to start a company. You know, Roy will just want to help you out. And I never listened to my dad until at some point we were really struggling, and we just called up Roy, and now he became our chairman. And we get to, you know, live and learn from him every day. So I think that the Greek theme in our industry is huge, because one of the guys who literally created, maybe the guy who literally created our industry, was a great first generation Greek immigrant role model who dramatically influenced my life when I didn't know him, and has changed my life for the last 25 years since he's been my chairman, and I've worked with him side by side. And I think that after the first generation of Greeks who were inspired by Roy in our industry, I think we now have the next generation who were also inspired by Roy. I hope also maybe inspired by a little of Roy's legacy, people like me and others, to maybe now take it to the next level. So I'm going to turn it over right now to Christos, but Christos is, you know, he's the future. I think that we have built with Roy, but with a lot of other contributors, a great company at Regeneron over the years with power capabilities and technologies. But you're not about what you've done. It's about, you know, what you're able to do now and where you're going to take it in the future and how you're going to change the future. And just like I think some of us Greeks have done it in the past, there's a real opportunity and a recognition that we have some incredible young Greeks who are really already changing the face of the modern biopharma industry by what they've done. But more importantly, they're going to take us, I think, to great new heights. And Christos is one of the top examples of that throughout the industry. As you've already said, this is the man who has already brought a cure for one of the hardest, most lethal diseases of our times that's legendary, that was in the movies, Ebola. And now, as you said, he's taken on one of the great challenges of our times, the COVID epidemic. So Christos, I think, is the next in the line of Greeks in our industry. I'm very proud of him. I'm particularly proud because he harkens from a town about 50 miles from my town in northern Greece. We're Greek Macedonians, particularly proud of that. And I think that he's a guy who's really already made an impact, but has a real chance, not only with COVID, but a lot of the other things he's doing, to take our company, but our industry to a whole nother level.
Wow. Christos.
Thanks, George, and thank you for the invitation. I'm very happy to talk to you today. So my background is in many ways very different, and in other ways very similar actually to George's background. So I was actually born and raised in Greece, in Kozani, yes, a very small town close to Kastoria, okay, where George's family is coming from. So there are two cities very, very close to each other. I was born and raised in Kozani, and then I went to Thessaloniki, the big city, the second largest city in Greece. And I went there for my college studies. And then when I was there, I was really interested in the research, okay? And you quickly realize that in order to do research, you need proper resources. And that's exactly what brought me in the US back in 2004. So I moved, my flight to New York was the same day that the Olympic Games actually started in Athens. Okay, so the day that everybody was going to Greece, I was leaving.
I actually went on that day. I probably passed Christos in the airport because I went with my father to the Athens Olympics.
So we probably met each other at the airport, but we didn't know each other back then. So, and I came to the US in 2004 to start my graduate studies at Columbia. And, just like George, of course, my father was always questioning all my choices in life. And one of the things that he was questioning was my choice to come to Columbia. Also not very well educated. He didn't know if Columbia is a good school or not. And during my first year at Columbia, there was an article in a Greek newspaper about Roy, okay? It was Easter Sunday, and there was an article about Roy and how he's inviting his friends at his house in New Jersey to celebrate Easter there together. And the article went on to say about, you know, his history and his legacy, of course. And part of his history was his studies at Columbia. So that was the first time that my dad told me, oh, maybe it's a good school, and maybe, you know, you can be just like Roy one day. Okay, so there are maybe some good people that can come out of Columbia. So of course, at the time, I have to admit, I didn't know who Roy was. And I also didn't know other people and other companies that were mentioned in the article, including the Regeneron and George, since Roy at the time, back in 2004, it was at the time when he was moving to Regeneron. So it was a very, you know, weird turn of events, I guess, that I found myself at Regeneron in 2011. And I've been here over the last 9 years, and I've been, you know, greatly enjoying my time with, I guess, the previous generation of Greeks like George. It's been an honor to work with them and utilize all the technologies that, you know, these guys have built over the years and try to develop all the drugs we've been developing. It's been a phenomenal experience, and, you know, I hope we can continue that success for many years to come.
Well, that's a wonderful segue into the extraordinary work that you're doing now, both of you. Chris Goulakos, maybe you can lead off with some questions, and maybe we'll get into yesterday's press conference as well, but by Regeneron. But Chris Goulakos?
Sure. And before we get into that, I'd love to just quickly walk our listeners through a quick overview of the Regeneron journey to highlight how remarkable this company truly is. So 32 years ago, in 1988, two MD PhDs with zero business experience decide to start a little company in upstate New York. 3 years later, you IPO, raised $91 million at a market cap of about $300 million. It then takes you 20 years before your first drug is approved by the FDA, and 25 years before turning profitable. And now, in 2020, Regeneron is at the tip of the medical spear in the combat for the corona cure. Dr. Y, how did those first 20 years lay the foundation for the enterprise and the medical and commercial success you've achieved since?
Well, that's a great question, Chris. And you did summarize it well, brought back some painful memories, but no, they were all great. They were all great memories. And I think that back then, actually, for the first 20 or 25 years of our existence, we were literally criticized and slammed by the investment in the analyst community constantly. And we were cited as the example of why you don't want a bunch of scientists running a company, because we didn't know how to make any money. And obviously, we've dramatically turned it around in the last 5 to 10 years. But as you said, it was really, the success really was dependent on the investment that we made for those first 20 or 25 years, during which we didn't make any money, that have laid the entire foundation for our success today. And I think there's an enormous lesson to be learned there. It takes a long time to truly innovate. It takes a long time to truly change the way people do biology and invent new drugs. And somebody has to be willing to put in that time and those resources and that effort. And we did it. And not too many other people right now are actually doing it. And I think there's a lesson to be learned there for society. I think that some of the biggest challenges we have that now face mankind demand those sorts of investments long-term in building innovative technologies, whether it be in all these disease areas that we're still all challenged with, or whether it be trying to address other existential threats to mankind like climate change and so forth. They're not going to just happen by policy. They're not going to happen by politicians, arguing over things. They're not going to happen by something quick happening like this. History shows that it takes enormous investments in time and resources to build truly new capabilities that nobody else has that can take you to that next level. And I think that I'm hoping that this coronavirus epidemic will be a great example whereby maybe more people can actually see that in order to create the power and empower scientists to have the ability to save the world by coming up with cures for a pandemic, you had to have put in the time and the resources over decades to create those capabilities and technologies. I believe there's nothing more important for our society and for humanity than to invest deeply in innovation and in long-term research capabilities. I think that we're a proven example of that. And I'm hoping that we can demonstrate, that Christos can demonstrate, that by using these sorts of approaches, we can make an impact when the world needs it most.
Great. And do you mind providing a layman's understanding of corona, how it compares to SARS, Ebola, and what the response strategy that you've undertaken as far as repurposing old drugs versus developing new antibody cocktails?
I think I'll turn that over to Christos. Maybe he can talk about that.
Great.
So both coronavirus and Ebola virus and MERS virus and all these viruses that, we've worked in the past, they are, you can think about them as very small particles that in order to both replicate and also cause disease at the same time, they have to get into one of our cells, okay? So they cannot replicate on top of, the table or on top of any surface or anything like that. In order to do that, what the virus does is it needs to attach itself to the cell. So there is an interaction between the virus and the cell. And this interaction is required for the virus to get inside the cell and start replicating. So when we, you and I get infected with any virus, or when we get vaccinated, for example, our body starts making these molecules that are called antibodies. And the role of these antibodies is to block the interaction between the virus and the cell. So you can think about it as a molecule that goes in between the virus and the cell, blocks this interaction, and then the virus basically becomes unable to get inside the cell and replicate. Okay, so that is this is something that is required for all viruses. So every time you're vaccinated or every time you are infected, your body basically creates these antibodies, and these antibodies are required for protection from subsequent infections, but also required for curing the infection when you start getting infected with one of these viruses. So what we are trying to do here for all of these therapeutic programs, both for Ebola and now for coronavirus, we are trying to make in our humanized models that we can go and talk about in more detail, these fully human antibodies that are going to block these interactions. And basically, they're going to, what we call, neutralize the virus. They're going to make the virus unable to get in the cell and replicate. So that's exactly what is happening naturally in our body. Okay, that's exactly what is happening with vaccines. That's exactly what we are trying to reproduce and provide to all these patients as drugs.
Great, and take us in the lab. So you have Velocimune, which is you've taken human genomes, transferred them to mice, and that is how you're testing these antibodies?
This is actually how we are making these antibodies. So, and to George's point, this is one of the foundational technologies that Regeneron is using now for all the drugs that we currently have in our pipeline. So there are tens of different molecules that came out of these Velocimune mice. So what these mice are, they are basically mice where all the genes that are normally encoding for antibodies have been replaced with the equivalent human genes. So these mice, they basically are just like a normal mouse, but the immune system of these mice is identical to the human immune system. So you can think about it as mice where there is a mini human immune system inside each one of them. So just like I said before, when we as humans, when we are infected or vaccinated, what we do is we make antibodies against all these pathogens. So we can mimic this whole process in a mouse. What we do is we challenge these mice with our favorite virus. In this case, what we are talking about today is coronavirus. And these mice are going to be making human antibodies that are indistinguishable from the antibodies that a human being is going to make. And what we are doing is these mice are going to be making thousands and thousands of these antibodies. We can take these antibodies out of these mice, and then in the lab, we can test each one of them, each and every one of them, and we can identify the best antibody at blocking exactly what I was describing before. There are some antibodies that are going to be very bad at blocking this interaction between the virus and the cell. There are some antibodies that are going to be phenomenal at doing that. So we want to go from thousands of antibodies, and we want to select the few, a handful of antibodies that are going to be the best at blocking the virus from entering inside the cell. And eventually, these are going to be the best drugs that we can make to become these antivirals and prevent disease or cure disease. This is what we did for Ebola. This is what we are doing now for coronavirus. But also, these are the same technologies we are using to make antibodies that can cure cancer. We can make antibodies that can cure autoimmune diseases. And we have several of these drugs are among the drugs that George developed over his career and are now FDA-approved products. Some of them are still in clinical trials, and we hope they're going to become FDA-approved products. We have a whole panel of technologies, from immunizing the mice, getting antibodies in the mouse, isolating the best antibodies, putting them in specific cell lines to produce lots and lots and lots of these antibodies so we can scale up and have enough material to be used in humans. Every single thing are multiple different technologies that we developed over the years, and we put them side by side so the whole drug development process can happen so fast and so efficiently.
Great, and Dr. Wyatt, why don't you take us into the boardroom? It's late 2019, reports of a novel virus start emerging in the headlines. What's the initial response, and how do you mobilize Regeneron to prepare for the new normal?
Well, to be honest with you, it didn't really happen in the boardroom, and it didn't really happen at the highest levels. We were following the news like everybody else. And it was really probably at some point in January where, we started being concerned about this, that maybe this was going to become a problem, maybe this was going to become a problem that was actually going to cross the shores and hit America. Though I have to admit, it was hard to imagine the devastation, especially here in New York City, which is, you said that we're at the epicenter of the response. New York City is at the epicenter of the crisis. So it's hard to imagine, and I never imagined the devastation that it would wreak. But in any case, when it started becoming more obvious, at least to me, we went to our head of infectious disease, Christos, and we said, "What do you think, Christos?" And he says, "Yeah, I've been following this, and this, let's hope that this doesn't develop into anything. But we're following it, and we understand this, and we've already started to synthesize the DNA of this virus to start essentially doing exactly what he described, to start immunizing mice so that the mice, before you ever deal with a human being, we can actually essentially challenge the mice with this virus. The mice can respond. We can clone out their response, their antibody response, and be ready to start giving it back into humans." So it didn't really happen at the boardroom. It happened, at Christos being ready and prepared for us. So by the time we went to him, he was already on this. And that together with, all the people he's collaborating with and all the capabilities and technologies that we built over the years that allowed him, as we said, in record time to now mount this new potential drug, this new potential cure against the coronavirus and bring it into humans. So he set the record for Ebola a few years ago. It took 9 months from starting the challenge and starting the project to going into human trials. So now we're counting on Christos to break his own record for coronavirus, in that within about 5 months of him starting the project in January, we will be in clinical trials in human patients. And these antibodies that Christos has already isolated, and he was able to isolate them from 2 related technologies. He was able to isolate them from these genetically humanized mice, but he also adapted or used adaptations of the same technology to get them from humans who actually recovered from coronavirus. You've heard of convalescent serum. Well, he's literally cloned the antibodies in convalescent serum, but only the best ones, as he's described. He took all of these thousands of antibodies. He took the best ones, and he's already working with other team members throughout Regeneron. They've already, it's called scaled it up. They've already started manufacturing these giant bioreactors, and we're purifying them. And by June, these potential cures for coronavirus will be in human clinical trials, and in 3 kinds of clinical trials. One is, as you would use a vaccine, to protect people who have never been infected. So it's a prophylactic preventive vaccine. So we're going to be going to one trial to prove that it's preventative, just like a regular vaccine would be, except here you don't have to wait or count on your own immune system to respond to the vaccine. You're being given the product of the vaccine, that is these powerful antibodies. So we are very hopeful that Christos' antibodies will be able to protect people who are at high risk, healthcare workers, first responders, or the elderly, and so forth. We have a lot of hope that they will protect these people and prevent them from being infected. We're also going to people who are early stages of infection. This is what Christos did with Ebola, and he showed even for Ebola, which is a much more lethal, I mean, obviously, coronavirus is much more widespread, but Ebola is a much more lethal disease. It used to be universally fatal. If people were given Christos' antibodies at the early stages of Ebola, when otherwise they would die, he was able to save over 90% of them. This is from Ebola. So we're also very hopeful and have some confidence that when we give Christos' antibodies to people in the early stages of coronavirus, we will prevent those people from developing severe disease. And that's the second clinical trial. We're doing all these simultaneously. The third clinical trial is we are going to those patients who are already sick and on ventilators and so forth in the hospital. That's going to be the toughest population to treat. But even in late stage patients with Ebola, the same approach that Christos used to make antibodies in the Ebola was able to save a large number of those individuals as well. So 3 trials: prevention, which we're hoping it could be almost universally effective; early treatment, where based on the Ebola, you think that you may be able to prevent the vast majority of these patients from progressing to serious disease, but then we might even be able to help some of the late stage patients, the ones who are the sickest, who unfortunately are in the most dire straits, who are on ventilators and so forth.
Chris, one more question maybe from you before we take questions from the audience. We're actually, through you're clicking on Zoom and Facebook, you can click. We're taking questions from people now, so Peter and I and Chris will do that. Chris, you want to one more question, and maybe we'll turn to some of the audience questions.
Yeah, sounds great. So while we're compiling, maybe one last, thinking of the future, how do you see the medical industry adapting and evolving? And how does the social fabric get back to a new normal?
Christos, you want to take that on?
So, what we've been discussing a lot is we hope that this is going to be a turning point for both our industry and society. And I think that kind of relates to a few things that George said at the beginning of our discussion, and also some of the questions that are being asked. I can see them on the notes here. So for, something that is now very, very obvious is that disease, is obviously one of the many, many important problems that society is facing. And unless you do investments on research and technology development and drug development, then you're going to be facing yourself from time to time with, threats to life and society and how we live and what we call normal, probably all the time. And being better prepared for all these things is, I think, necessary, right? So, and I think we just learned a very hard lesson over the past, few months. We hope that it's going to be a lesson and we're going to actually learn something from it, so that next time something like this happens, we're going to be better prepared and have all the technology in place to more quickly respond to it. And, so that's something that I think that is going to be a turning point for society. Now, and of course for our industry as well. So I think over the past few months also, we've seen a lot of focus on the research and the development of new both vaccines and therapeutics that are being done by both Regeneron and other companies. So everybody's looking at us to come up with solutions, right? So overnight, we became from the bad players in the healthcare system to the ones that everybody's looking for the magic solution that is now going to solve the coronavirus crisis, and everybody can go back to their normal life. So hopefully, we can prove, something that should have been obvious, but maybe it wasn't, that, we can provide obviously a lot of value, right? And, solving all these threats to society like disease, and it's not just the coronavirus, we can think about cancer, we can think about a lot of these neurodegenerative diseases, right? So there are so many things that we are going to be facing more and more often as society, and it's up to us to make sure that we develop the right therapeutics and develop them the right way so that they are available for everyone that needs them, and make sure that we can continue going on as a society. So I don't think it's just a problem for coronavirus. Hopefully this is going to be a short-term problem that we're going to quickly solve, but we're going to face other infectious diseases and many other diseases that affect, that are coming from a lot of these biological systems in the future. There's no doubt about that. And, our work and our effort is going to be required, and hopefully we're going to be able to deliver for both the coronavirus and all the other things in the future.
Yeah, let me add to that, by the way. I believe in everything Christos just said. And the other George, maybe the other guy on this panel right here, who remembers the 60s, but I'm an old guy. And the world was very different back then. And it was, I think, a much different time to be growing up. Science and technology was highly elevated in our society. The presidents at the time were challenging us to use science and technology to solve all of mankind's problems, all of the existential threats facing mankind. And so I thought it was a heroic choice to become a scientist and to take on these challenges. And I was just saying that I think that society has sort of lost that positioning of science as something that's so esteemed and so valued, but we've never needed it more. We truly face existential threats as a species, and I truly believe that our only salvation is to invest more in the one thing that distinguishes us from every other species, the ability to use our minds to think our way out of our problems and to how to come up against these challenges and solve them. And I really do hope that this is, as Christos said, this is maybe a wake-up call, but also a time when, I've been so frustrating following politics, following the news cycle, following everything over the last many years now, where we get into partisan politics, we start arguing and talking about what I think are useless details and distractions. We got to become united as a species, as a humanity, to recognize that we have real problems, we have real threats. They're going to only come more fast and more furiously, like this epidemic. And we have to come together and we have to focus on being ready and being prepared against these real enemies that threaten all of us. We are one species, we're one humanity. We have to be united. We have to stop fighting amongst ourselves. We have to come together. And I do think we have to, re-energize ourselves and refocus on this challenge of using science and technology to save our species. And I hope that this will be a great example. I hope we come through, or one of the other great companies in our industries come through and really, saves the world here. And I think that then the world recognizes that this is what we're going to have to be doing more of, not less of, and avoiding the silly arguments and instead focus on the really important things, the really important challenges, and how to come together to solve them.
So I'll lead off with the first question from the audience, George, if I can, and that is, does this demonstrate American ingenuity compared to other efforts around the world? How would you rate American ingenuity? And the quick follow-up to that is, has the role of government here been a positive one, and what kind of grade would you give it?
Yeah, I think these are both great questions. I am a big believer in American ingenuity. I think, like I'm sure many Greek Americans, I was raised to be incredibly proud of Greece and my heritage, but I was also raised to believe that the people who came to America, there was just something special about them because they were in some way self-selected to be yearning for more and to be exploring more and to be asking more questions. And, you know, Christos is just another great recent example of that. Somebody who recognized that in order to achieve everything that he wants to achieve, he has to cross an ocean in order to do it. And I believe that what makes America special is truly been selected and is our genes. And history has proven it. Over the last 100 years, there has been no other society that has been as ingenious and as inventive and as innovative. We are at a dangerous time, though, in our history, in that we may be going away from that. The incentives, the arguments that I hear about from politicians about where they want to direct resources and so forth. American ingenuity and inventiveness and technology has led the world for the last 100 years. And we should be committed. This relates to what I was just saying before. We should remain committed to making sure that that remains our competitive advantage and that the world actually needs us. It's not only in the case of this COVID virus, couldn't have been for Ebola, but it could be for almost any disease. The drugs, but also the new technologies historically have always come from America, usually from first and generation immigrants living in America, many of them Greek, but it happens in America because of our special environment, because the special people that decided to come and live within these shores. So absolutely, American ingenuity is, I think, very special, but we have to recognize that we cannot take it for granted. It is in our freaking Constitution. Our founding fathers, led by Ben Franklin and Thomas Jefferson, put into the Constitution that we should do whatever we can to incentivize inventiveness. They actually had in the Constitution the fact that if you're an inventor, we want to encourage you. How? By giving you a patent for your invention. So you will be able to profit from it for a certain period of time before it became then free to the world, because they literally recognize, our founding fathers, that that feature, that ability to patent your invention and profit for it would be a great incentive and a drive for people to be so inventive. So I think it's a combination of our people, but also the vision of our founding fathers to actually encourage in our very Constitution the incentives that would drive the most innovative behavior on the planet over the last couple hundred years. And we can't lose sight of it. That's what I'm saying. We have been the leaders in the world in terms of innovation, not only for drugs, but across all sectors. And we can't take that for granted. We have to work as hard as our founding fathers to make sure that we immortalize the incentives that are necessary to drive great thinkers, great inventors to want to do that and to be incentivized to do that.
George, I hope we can find a way for every young person in America to listen to what you just said. It's extraordinary. Peter, do you have a follow-up question or two that you're getting? I know there's all kinds of questions coming up on the screen. Peter, do you want to ask a final question or two?
Peter? I think you're muted.
Given, I just read a question that I think is appropriate given what Dr. Yancopoulos just said. And this is from someone from Vasiliki in New York. The question is, since Greece has a tremendous amount of engineering and scientific talent, what can Greece do to play a vital role in the world of research and innovation?
Well, I'll start by saying that Greece already is a great source of worldwide ingenuity. Some of our top scientists come from Greece, like Christos, but he's a person who left Greece. Maybe he can speak to the notion about, you know, what Greece can do to not only feed great brains and great young people to the rest of the world, but what Greece can do on top of that.
Yes. The reason why I personally left from Greece and the reason why a lot of people are leaving from Greece is because there is not a lot of investments There's not this environment there to allow for all these investments, for this long-term planning, long-term innovation, long-term technology building that will eventually create all these companies like what George was describing for Regeneron. This is a very long-term vision. These things do not happen overnight. And unfortunately, this vision has been lacking in Greece over the past several years. So this is an effort that at some point will be great to start, but everybody should realize that it's not going to happen overnight. This is something that can start today. It's going to take many years, and eventually it can build an environment that can allow for this innovation. And hopefully then all these people are going to stop leaving the country, but are going to, stay in the country. I think I'm a very bad example, unfortunately, of Greece's investments. As you all know, education in Greece is totally free. So my education, both my all the way actually including college, was paid by the Greek government. So I'm in theory an investment of the Greek government, and now I am contributing somewhere else. So I think it's a great example of how an investment of Greece is actually not paying off. And in order for people like me to stay, they need to encourage an environment that's going to allow for all these people to stay.
Peter?
I have another question from Theo in Connecticut, which says, are you looking to see if COVID-19, in addition to affecting the lungs, causes other physiological effects, for example, such as COVID-inducing deficit of protein E and S, which then can cause blood clots, as has been observed in many COVID-19 patients?
No, that's a great question. So absolutely. We and others now are trying to understand the pathophysiology of the disease. As you probably know, one of the drugs that we currently have in clinical trials is actually something that we are repurposing from other uses, something that we use for rheumatoid arthritis. Is an anti-inflammatory drug. And the reason why we are using this is because it has been observed in people that there is inflammation in the lungs, and the inflammation is the major cause for the requirement for supplementation with oxygen. So we believe that this drug can reduce the inflammation, especially at late stages of diseases, and eventually these people can leave from the hospitals earlier. So similar things, people are trying to understand the pathways that are involved in either the clotting, okay, what is contributing to clotting, what is contributing to tissue damage, how the virus is actually causing disease in the human body. And the more we understand about the disease, then we can use these specific blockers, like things that block either the clotting or the inflammation that I mentioned, to not exactly directly block the virus, but block the virus from causing disease.
Very interesting. I have hundreds of questions, but I think I'll ask one more, which, and this comes from John in Chicago. So pathogens have come and gone in world history. Is this just the natural order of things, or is coronavirus a special situation?
I'm gonna comment first on this, and then I'm going to turn it over to Christos for the details. But I do think that, and I've used this analogy before, and I think it's actually true and relevant. I think that we could look at our species on Earth no differently in many ways than bacteria growing in an Erlenmeyer flask. I'm sure when you were all in school, you might have ever grown bacteria in a little flask, and the bacteria grow and grow and grow until they become so concentrated that they literally kill themselves off, okay? What we have to recognize that we're doing exactly the same thing. The density of the population is increasing, that it's gonna result in two things. Naturally, just like bacteria in an Erlenmeyer flask, the poisoning of our environment and the increasing spread of diseases due to the increase in population density. So I don't think it's an accident, nor that it'll be a rare event, okay, that there will be an increase in transmission of infectious diseases. It just has to be the case because of the increases in population density. So I'm not saying that there's necessarily anything special about the coronavirus, but there is something that we have to recognize, that these threats are becoming much more profound across much larger swaths of the population just because of how the world has changed in a couple hundred years. If there was a new virus that broke out in Wuhan, China, I'm not sure Wuhan even existed 200 years ago, but the odds are it would never have made it to America. Okay, now it's inevitable that it made it to America long before we even knew about the Wuhan virus. Now the evidence suggests it was already in America. And so that's one thing that we have to recognize, that the world has changed. We really are on the other end of our growth cycle, and we need to do something about it very, very fast and very furiously, which to me means an increase in all sorts of investments in engaging the best and the brightest minds to take on these problems. So I don't think there's anything special about the coronavirus. I think that what's really, the problem is that society and mankind and population density and globalization have gotten to a point where not only has it had devastatingly negative impacts on our own Earth, but it's going to have increasingly devastating impacts on the spread and the rate of spread and the devastation to the spread of diseases of all sorts, including infectious disease.
Christos, a question for you from a Dr. Liakias. What do you think of the status of those who have recovered from COVID-19? Do you think that those people will maintain immunity, and for how long?
No, this
And no, that's not Scott Gottlieb asking. That's a Dr. Liakias.
That's a doctor who just recovered, by the way.
No, that's a fantastic question, right? So unfortunately, we don't know yet. Okay, and we hope that this is going to be the case, of course. So as we mentioned before, everybody who has recovered from infection, they should probably have their own antibodies. They made their own antibodies now, so they should be protected from subsequent reinfection. So we are actively monitoring what we call the titer. This is the amount of antibodies in somebody's body and how this correlates with protection. The rule in infectious diseases is once you get infected with something, you're protected from it for a long duration of time, sometimes for the duration of your life. And we have to see. We hope that this is the case here. We have to wait and see.
Okay, a question for you.
A quick follow-up.
Yeah.
Thoughts on second and third wave risk and the trade-off between reopening the economy too soon and the collateral implication from a medical front?
Well, this is definitely, the balance, right? So that society is facing. We hope that our job here is to develop therapeutics fast enough so that these therapeutics are going to be available for the next wave, if there's going to be a second and third wave for the coronavirus. There is a trend now, both here in New York, elsewhere in the US, and definitely in Europe and in Asia, that the number of cases is going down. This is fantastic. Of course, it was done by, a lot of people staying at home and closing the economy. But this also allowed us, they gave us the time that we need to develop these therapeutics that hopefully over the next few months, we're going to know if they work or not. So our job here is to be prepared. So if there is another wave come, the fall, the devastation for the healthcare system is not going to be as pronounced as it was during the first wave.
Dr. Yancopoulos, Dr. Kyratsous, this was in a very fast hour, and we don't want to keep you any longer from your very, very important work. We don't want to wear out our welcome, but I have to tell you that this is about the most inspiring hour on TV that I've had in several months. And if we can broadcast some of this out to the world will be a better place, not for hearing from you, but for all the work that you do. So on behalf of the Hellenic Initiative again, and all of us on today's program, thank you. Ευχαριστώ πάρα πολύ from the bottom of our hearts, and thank you for everything you do. Thank you, George. Thank you, Christos. And thank you, everyone who was listening. Tune in again in 2 weeks for the next one. Thank you, everyone.
Everybody be safe.
Thank you, Artigo.